The overall goal of this procedure is functional characterization of in vivo tissue by combining high-resolution structural information with quantitative spectral data.
This method can be used to answer key questions in the field of cancer biology, such as quantifying the effects of therapy on tissue microstructure and perfusion.
The main advantage of this technique is that a single probe can be used to collect both image and spectroscopy data from the same location.
The implications of this technique extend towards gastrointestinal cancer therapy because the probe is compatible with conventional endoscopy techniques.
Generally, individuals new to this method will struggle because small irregularities in probe placement can significantly affect data accuracy.
We first had the idea for this method when we realized the importance of combining a high-resolution imaging technique with a quantitative spectroscopy method.
Visual demonstration of this method is critical as the preparation steps are difficult to learn because of the complexity of both the construction and calibration.
To begin assembling the high-resolution fluorescence microscopy modality of the multimodal microendoscope, mount a 470 nanometer dichroic mirror in a 30 mm cage cube. Attach cage assembly rods to the front, right, and left sides of the cage cube. Attach a stress-free retaining ring to a threaded cage plate and screw a lens tube into the retaining ring. Attach another cage plate to the lens tube aligned with the first cage plate. Next, secure a UV-enhanced aluminum mirror in a right-angle mirror mount.